Hello everyone, my name is Alexandra Moreira. I'm a SURP student working under Dr. Scott Argraves in the Department of Regenerative Medicine and Cellular Biology. I'm a biomedical engineering student at the University of South Carolina. This summer I worked on a project studying the effects of static mechanical strain on living tissue engineered fluoride constructs. Now, just to kind of give you an overview of like the idea behind this research, kind of give you an overview of the different types of blood vessel replacements that are out there today. The optimum option would be autologous grafting, and that basically just means taking a graft of an artery from a different location in the body and replacing it to the diseased or affected artery. And although this is like the optimal option, there are some cases with patients that there's a limited amount of eligible patients. So I guess patients with pathological diseases such as atherosclerosis and other cardiovascular diseases aren't able to be eligible for these types of surgeries and for this option. And there's also have been seen to have many complications with grafting, including compliance mismatch and insufficient mechanical strain. And that basically just means when you're trying to replace an artery, a major artery in your body, and your only option, I guess, is to choose a vein, you know, there's many mechanical differences in doing such surgical procedures. So they would provide insufficient mechanical strength for the patient, and which would lead to grafting failure. Also, there are synthetic vessels available, and these are primarily composed of biomaterials. And although they are easily manufacturable and readily available most times, they have many biocompatibility issues, including high thrombosis and inflammatory responses. Then there's tissue engineered blood vessel constructs, and this is these constructs are composed of biological materials, including human vascular cells and extracellular matrix. And the key characteristic with these types of constructs as an option for replacement grafting is that these have tunable mechanical properties. So you can actually adjust the mechanical properties in the fabrication process of making these blood vessel constructs by adjusting things like composition and culture environments. So these are really important characteristics of these constructs. And here's just a slide just to kind of give you a general overview of biological components of a blood vessel. And so the three components that contribute to the mechanical response of a blood vessel are the elastin, collagen, and smooth muscle cells. Now, elastin is found in the medial layer of a blood vessel. Here's the lumen, as you see, as a reference point. And elastin is responsible for the passive mechanical response. And collagen is located in the adventitial layer surrounding the fibroblasts. And it is also a passive, it is responsible for the passive mechanical response of a vessel. And the smooth muscle cells, interestingly, are actually responsible for the active mechanical response, while a blood vessel is going under vasodilation and vasoconstriction. So I'm just going to kind of talk about the importance of elastin in our studies. Elastin is an extracellular matrix protein that confers mechanical elasticity in a vessel. Now, it is responsible for the low pressure circumferential mechanical response in a blood vessel. It is also a passive flow-bearing element in a tissue and provides structural support to a vessel while it's undergoing cyclical strain. And it is used biomechanically to model a nonlinear elastic isotropic material, which basically means the isotropic material, the mechanical response does not matter what direction it's going into. And so for collagen, it's also an extracellular matrix protein that dictates the tissue mechanics at high strains and high physiological pressures. Collagen fiber alignment is oriented axially and circumferentially around a blood vessel. Now, collagen is used to model the nonlinear and isotropic material, so the direction of the mechanical response does matter. And so, kind of talking about the rationale for my study would include that elastin and collagen are fundamentally important for vessel material and biomechanical properties. Now, in a study that Dr. Argraves and I read from Dr. Wagenseal's group, showed that elastin production results directly from mechanical stimulation during the developmental stages of blood vessels. And this is due to stretching of the actual vessel due to hemodynamic forces and due to increased cardiac output as development progresses. And so, the idea is for fabricating vascular constructs, applying mechanical forces will affect the production of elastin and collagen, thereby influencing the biomechanical properties. And so, doing studies of how to vary these forces, how varying these forces affect biomechanical outcomes can be used to learn how to fabricate constructs that have optimum biomechanical properties. And so my project goal was to determine experimental conditioning parameters that influence mechanical and histological outcomes of these constructs that we will make. And this is on these two parameters are on two levels. So the first one is changing the culture environments and seeing their effects on the biomechanical and biochemical properties. So we use vertical post and Argraves molds as our two different culture environments. And we also tested different levels of static strain. So static strain meaning different stretch ratios. An unstretched setting at the stretch ratio of 1 versus a stretch ratio at 1.5 being the stretched strain level. And so our ultimate goal with all this research is to fabricate elongated tubular tissue constructs that have optimized mechanical and material properties similar to those that are contained in native tissues. So, getting to my experimental hypothesis, we believe that we hypothesize that mechanical and biochemical properties of constructs are influenced by culture conditions, in this case, mold versus vertical posts. And also, by increasing static stretch, we will improve the mechanical, morphological, and biochemical properties of these constructs. Now, my first experimental aim included performing progressive static mechanical strain testing on tissue-engineered tubular toroid constructs. Here's a kind of a picture of, I'll get more into detail in the next few slides. You'll understand how we actually make these toroids and how we actually stretch them. So my experimental plan for quantifying these properties include biomechanical testing, where we subject these toroid constructs to tensile testing. And with tensile testing, we are able to investigate the response of collagen and elastin by calculating their intrinsic elastic moduli. And also to track the changes in morphology by stretching these toroids, taking them from their molds and putting them around these vertical stretching posts, we analyze the differences in the toroid dimensions using outer diameter, thickness, and inner diameter information. And we also backed this data up. We confirmed this, the mechanical response with biochemical data. So we measured, using real-time qPCR, the messenger RNA expression of collagen and elastin at day 12 of harvestation. And we also used histological staining in order to qualitatively see the distribution of these extracellular matrix proteins within these toroids. And so, getting to the fabrication approach that we use, we cellularize these cultispheres, which Katie mentioned earlier, they're actually gelatin beads that served as a scaffolding system for these tubular constructs. And we seeded them with a 1-to-1 ratio of human umbilical vein endothelial cells and vascular smooth muscle cells. Now, these macroporous microcarriers are seeded, and by the end of development, as you can see here, these circular these little circles here are actually the cultispheres themselves, and the interspaces in between them are actually the extracellular matrix that comes out of the cultispheres and forms a structure, the tubular structure. So this right here is a toroid, and it's kind of a donut-shaped tubular structure. And so how we actually make these toroids? So we have a mold template here. We submerge it in Argraves, we let it solidify here, and we wait 15 minutes and we take it out, and we're left with this mold where we can then load these molds with the cultispheres that have been equilibrated in a spinner flask after several days and grown in a 6-well plate to make these toroid constructs. Now, just kind of the overall experimental design for my project was we had 2 different culture conditioning settings. So one was the Argraves mold culture conditions, and as you can see here, and the other one was static strain via vertical post stretching. So we had one post with a 2 millimeter diameter, which would represent the unstretched condition, because these toroids were fabricated and designed to have an inner diameter of 2 millimeters. So by placing them over this vertical post, it would be unstretched. And we had a 3 millimeter in diameter vertical post, which would be the stretch setting. And that would be a stretch ratio of 1.5, and this would be a stretch ratio of 1. And here's kind of a visual aid of what I'm actually talking about. So we're taking these toroids out of their molds and placing them onto these vertical post stretchers. And to kind of give you an overview of the actual process involved with this is we have these cultispheres that are seeded with these cells in a spinner flask for 5 days. We take them out of the spinner flask, we load them into the molds, we let them culture for 2 days, and after 2 days we put them onto these vertical stretching posts at those 2 different, so 2 groups of toroids at these 2 different stretch ratios, one being stretched and one being unstretched, and we let them sit there for 5 days and under this strain conditioning. Also, we have just the cultispheres in the spinner flasks after 5 days, directly putting them into the molds and having them stay in the molds for a total of 7 days. So these two time points are at day 12, and so I'll be comparing the data with biochemical and biomechanical properties later on. So after making these toroids, to get the data and to see, the changes in these properties, we subject these toroids to tensile testing. And here's kind of the machine that we use to do this type of testing. And here's a toroid mounted on to these 2 cannula here. And here's kind of a visual aid, a video of what actually goes on while the test is operating. So this toroid here is being stretched, displaced from 1 millimeter of displacement to 2.2 millimeters of displacement. And all those data points measure the force that the actual toroid is pulling on this cannula. At the bottom of this cannula, there's a load cell that measures the force in grams. And with all that information, you can calculate the stress strain curves, and from that, you can see the relationships with their intrinsic mechanical properties. So here are just kind of some fundamental engineering equations that we use to kind of characterize these properties. Here's just stress equals force over current cross-sectional area, and stretch ratio equals the final test segment length divided by the initial test segment length. And now, here is a graph of the total nonlinear elastic mechanical response of blood vessel, and of these toroid constructs, And so here's stress versus stress ratio. Now in this region, at low strains, elastin contributes to the low strain mechanical response, while at higher strains and higher physiological pressures, collagen contributes to that mechanical response. And so after looking at that data, we also wanted to see the changes in morphology like I mentioned before. Now these graphs, just to kind of summarize this slide, we did see significant changes in the morphology of these toroids just by simply changing their culture environment from the Argraves mold conditions to stretching them on vertical posts. We saw significant differences in each category of the outer diameter, thickness, and inner diameter. Now, on the biochemical side, we did see an interesting relationship here. So the messenger RNA expression of elastin actually decreased significantly by changing these culture environment settings. So by just taking them out of their happy agarose mold culture conditions, the coded message for mRNA went down when we placed them into the vertical post culture conditions. And for collagen, we didn't see a significant difference. And going back to the mechanical data here, so the elastic modulus kind of is a material property that describes mechanical response of these toroids. So it's simply by doing a regression on the separate portions on that graph that I showed you earlier. So here we see there was a minimal effect on the mechanical properties. This could be for several reasons, which I'll get back into in my discussion portion. But here, just kind of wanted to show you kind of qualitatively the distribution of these extracellular matrix proteins in the toroids. So here is a picture of a cross-section of a toroid, and these red circles here are actually the microcarrier cultispheres. So you can appreciate the extracellular matrix that actually comes outside and forms the actual tissue. So the blue represents collagen, and the red represents muscle fiber and fibrin. And here is hematoxylin and eosin staining that kind of just shows you the cellular distribution of these of the extracellular proteins. And so some of my conclusions. So the mechanical strain conditioning of these toroid constructs were seen to have minimal effect on the biochemical and biomechanical properties. And static stretch caused significant morphological changes in these toroid constructs. Also, the mRNA expression showed a significant decrease in elastin expression going from the agarose mold to the vertical post-stretching conditions. And just some kind of perspectives from the study. These studies clarify how culture conditions influence biochemical and biomechanical properties in these vascular constructs. Also, further studies are needed to optimize fabrication protocols in order to enhance material properties, and we can do this in many ways. By another environment could be cyclical strain, so a dynamic culturing environment, and also varying the culture duration via in the molds or on the posts. So there's a lot of variations you can do to actually optimize these characteristics. And also, information from these toroid experiments will allow us to model and predict the mechanical behavior of elongated tubular constructs, which is our goal, to take those elongated tubular constructs and replace them as replacement grafts for people who need them. And so some ongoing and future studies, we plan to fabricate and mechanically characterize elongated tubular constructs, and after optimization of fabrication protocols, after those are found, we will then subject these toroid elongated constructs to dynamic culturing conditions. So putting these elongated tubular constructs, which I'll just show you in a minute, in a bioreactor, subjecting them to pulsatile flow at physiological pressures. Now here's a picture of a successful elongated tube that we were able to fabricate. We will continue doing testing Here's another picture. We will continue to do biaxial testing, including inflation and extension tests, to better characterize mechanically these constructs, looking at burst pressure and compliance, and being able to characterize constitutive model for these constructs. Now, I would just like to take a minute for acknowledging the Department of Cellular Biology and Regenerative Medicine, my mentor, Dr. Scott Argraves. He's helped me a lot in, really mentoring me this summer. And Sandy Klatz, our lab technician, she's really helped with mentoring me as well. And all of the grad students, Kirthi, Marion, Waleed, Jamie, Barth, and Cindy Ginter. And I'd like to also thank the Department of Biomedical Engineering at USC, my former professor, Dr. Shazley, his grad students, Oren, Ian, Mo, and Rhett. And I would like to offer special thanks to the biofabrication program under the SURP program, and also the NSF grant that actually funded that program. I learned a lot this summer, and I look forward to continuing research next semester. So, thank you, and are there any questions?